Transcription is the process of creating RNA, specifically messenger RNA, from DNA. After transcription, mRNA will travel to a ribosome to be translated into amino acids that will code for proteins. DNA is used as a template for RNA synthesis. The DNA is unwound by an enzyme known as RNA polymerase. Unlike in DNA replication, RNA polymerase doesn't need a primer to begin synthesis. This is due to the promoter region located in DNA. RNA polymerase locates the promoter because it recognizes 2 points in the region, the -10 box, or Tata box, and the -35 region. This is possible thanks to the sigma factor in the holoenzyme. The holoenzyme is needed for template binding and initiation of transcription, but only the core enzyme is needed for elongation and termination. The promoter is located directly upstream from +1, which is where transcription begins. At +1, RNA polymerase begins unwinding the 2 strands of DNA. In transcription, one strand is the template for RNA synthesis, and the other is known as the coding strand. Corresponding bases are created from the template strand. The template strand will always be read from 3' to 5'. This is due to the fact that RNTPs, or ribonucleoside triphosphates, are always added from 5' to 3'. In other words, nucleotides are added to the 3' end. The coding strand is significant because the RNA that's created will have almost the exact same genetic code. The only difference between the two is that where the coding strand has a thymine, the RNA will have a uracil. The RNTPs are made into nucleoside monophosphates by the removal of 2 phosphate groups. This allows the formation of a phosphodiester bond between neighboring nucleotides. The phosphodiester bond is formed at the 3' end. RNA synthesis can't go on forever. Termination occurs due to inverted repeats of bases. Inverted repeats, when transcribed, develop termination hairpins. Hairpins form from base pairing within the strand of RNA. The hairpin interferes with RNA polymerase. It causes the polymerase to pause. Following termination hairpins is a string of adenines on the DNA, known as a poly-A tail. The RNA will have a corresponding poly-U tail transcribed. The bonds between adenine and uracil are relatively weak, which allows the dissociation between RNA and DNA. The RNA polymerase pauses at those bonds and termination of transcription occurs. Well, what happens if the -10 box is removed from a gene that codes for an enzyme to metabolize sugar? Without the -10 box, the RNA polymerase would not know where to bind to the DNA. Transcription would not be feasible. If RNA polymerase didn't know where to bind, the DNA strands wouldn't be separated and a transcription bubble would never be formed. Sugar would not be metabolized in the organism lacking a -10 box.